and she's from the University of West Georgia.
That's great. Hi everyone, welcome to my presentation. My name is Anna. I'm an assistant professor of computer science at the University of West Georgia. I've been there for about a year now, where I teach mostly computer science classes. But today I want to talk to you about the work that I did during my postdoc training. I was there for 2 years at the Icahn School of Medicine at Mount Sinai in New York. That's where we initiated this project, and I'm still working on it. I'm very fond of it. So today I'm going to show you results that have been published, as well as new current results that are yet to be published. So I will be talking about how to integrate heterogeneous predictive models using reinforcement learning. That's the topic. So modern biomedical data collection is generating exponentially more data. And this data, they come in a variety of formats. Okay? Sometimes systems biology and machine learning can help us generating predictive models from that data. But it's not always obvious or clear what is the most appropriate data set to use, what is the most appropriate view of the data to use, and even what predictive algorithm to use when training with this data. So one way to improve the predictive performance of these models is to build ensembles. Now, that's one popular way to enhance predictive performance. And because all these models can come from a variety of data sets, they're inherently diverse. And building heterogeneous ensembles will help boost the performance. Now, sometimes we may want to find a parsimonious subset of models, right? One that's smaller in size and that boosts the predictive performance. Think of something similar to feature generation, feature selection, right? Where if you leave out some of the constituents that are not so great, that don't complement that well the ensemble, you might just want to leave it out. And also, if you're dealing with a smaller ensemble, with something that's more parsimonious, right, smaller in size, it can ease potentially the interpretability of that model. You can go ahead and analyze it better. So this is actually a technique called ensemble selection, right? How do you select models into an ensemble? And we're addressing this problem in a novel way with reinforcement learning. So reinforcement learning is another branch of artificial intelligence that deals with training agents. So you have an agent that is left to roam free in an environment with a goal. It has an objective, right? It needs to reach a goal. In this example, we have the agent that tries to reach the golden mushroom, right? So the way it learns is by interacting with the environment. It gets reinforcement signals from the environment. And during these learning episodes, it picks up that it may have some obstacles along the way, right? And as it accumulates this knowledge, it knows in future episodes, in future behavior to avoid those places. Okay, so eventually, if it spends enough time in the environment, it will learn an optimal path, right? By exploiting whatever knowledge it's accumulated up to that point, and also exploring new unseen, untraveled paths in the environment that can potentially lead to the goal faster or better, depending again on how we set up that goal. So how do we do ensemble selection using reinforcement learning? Well, we still have a structured environment. We still have our agent, which is the ensemble selection algorithm in this case, and it's left to traverse this lattice. This lattice is made up of all the possible ensembles that one can create from predictive models, right? So in this case, for example, we have 4 predictive models. What is the best way to combine them? So the agent starts with an empty set, for example, and as it traverses this environment, as it spends time learning how to find a good ensemble, it will keep adding models, right? It starts in the beginning with an empty set, keeps accumulating models, and eventually would learn a recipe of how to add things and what is the best ensemble there. And sometimes we have to develop these strategies of how to traverse the lattice, right? How is the agent interacting with the environment, and what kind of reinforcement signals it gets back? And that is in terms of the reward function. So we have to formulate this reward function in terms of ensemble performance, which is pure predictive power of the models, or diversity. So it's very well known that to reach a good ensemble, a good predictive ensemble, the models, the constituent models need to be good, need to have good prediction performance, but also diversity, right? It's a very fine balance, and it's not clear how to reach that. So imagine like if everything was very good and predicting very well, you would not add any novelty, right? Essentially, you can go with any of the constituent models and call it a day. But sometimes if you add diversity into this, into the play, you can boost the performance because you're adding novel knowledge to the ensemble. So designing these strategies is pretty tricky. We developed several strategies. Some of them look at simple, sheer performance, and some of them also integrate diversity. There are other ensemble selection techniques that operate in the same type of environment, right? In general, those the standard or traditional ensemble selection techniques are heuristic. They follow some heuristics. They're ad hoc in nature, and they do tend to work well in practice, but it is more difficult to guarantee good prediction, good performance. So the results that I will show you today are from a target problem. We're looking at predicting splice sites in various organisms based on nucleotide positional match. So basically, we're formulating this as a binary classification problem, as a prediction problem, where we look at DNA sequences from publicly available data sets from 5 organisms. These are relatively large data sets, and we create 10 backed versions of the data and train 18 of the most popular prediction algorithms on them, and end up with 180 base predictors. So you can imagine that for 100 base predictors, the ensemble selection problem is very computationally intensive. The agent ends up looking at a space of 2 to 180 base predictors, so, which is pretty large. So these are the results. On the x-axis, we have the number of base predictors at each experiment that the agent is selecting from, right? So for example, if you look at 80 over there, that means that the agent for this experiment has 80 models or base predictors that it can select into an ensemble. So we do this increasingly to see how it varies with an increasing number of models, how the performance varies with an increasing number of models. On the y-axis, we plot the Fmax. So this is the predictive performance. And we use the Fmax because the problem that we're looking at has a very imbalanced distribution. Splice site is naturally imbalanced. It has a ratio about 1 to 100. So Fmax is a more appropriate measure of assessing the predictive ability. So in green, if you look at the green line, you will see the best predictor, the winner from each pool of ensembles, right? So for example, at 80 over there, the best one among those 80 models has a prediction performance of about 0.66. That's the green line. We also plotted the full ensemble. So how well, what is the prediction performance that we expect to get if we put all the ensembles together? And that is the red line right here. All the other lines are our ensemble strategies that we developed. The first 3, greedy, pessimistic, and backtrack, again, are search strategies that look only at the model performance. The last 5 incorporate diversity. So this is a framework that allows for diversity to be incorporated very easily into the search strategies. So overall, you can see that as you increase the number of base predictors, as expected, the performance does increase, right? The more models you have, the better the performance. And on the other hand, almost all of the RL-based strategies perform competitively, competitive with the full ensemble. Okay? Now, the interesting thing here is that we are able to reach that performance by using a substantially smaller set of base predictors, which is great for interpreting those models down the line, for possibly reverse engineering the biological process of interest or the study that we're looking at. So one of the strategies in particular is able to even surpass the predicted performance of a full ensemble, and to do so with about 1/3 of the models available to it. So in conclusion, I wanted to point out that these reinforcement learning ensembles and this framework that we developed, they are competitive in prediction performance to the larger ensembles that can be obtained fairly more straightforward, while being substantially smaller or more parsimonious. And also, if we incorporate ensemble diversity, and if that is measured appropriately, we can build even more accurate and parsimonious ensembles. So I invite you all to try out our implementation. It's been available on GitHub for the results that I've shown you. And we're also interested in testing our framework for other data sets, not necessarily biomedical data sets, and also for larger data sets. And since this is a very computationally intensive problem because we're looking at an exponentially large space, we need to also develop more efficient, possibly parallel implementation. This is not a very straightforward problem to be parallelized, but we're looking into that. This is something that we plan to do in the future. I would like to acknowledge my advisor at Mount Sinai. This is the project that I started there, and I'm very fond of, so I've continued working on it. It's been sponsored by an NIH BD2K grant. I also like to shout out for the Minerva supercomputing team at Mount Sinai, because we use the HPC cluster to conduct our computational work. And of course, the CS department at West Georgia for allowing me to continue my research. Thank you all for your attention. And I would also like to invite you to my poster on Monday evening. It's at the exhibit level. If you want to talk about details about my project, and I'd be happy to meet you one-on-one and discuss more about this. And I'm happy to take any questions. Yes.
Yes. First, thanks for this great conference. I want to know, could you please explain more the formulation of your program? Are you going to find the common features for supply-side across different species? So I'm wondering, yes, for your supply-side. Yeah. What's the problem you want to address with this machine learning approach?
So we are predicting splice sites in various organisms. So splice site is this AG dimer that is located at the junction between an intron and an exon. So only about 1% of the AG dimers are actual splice sites. The rest of them are just decoys, right? Just occurrences, random occurrences of AG dimers across the genome. So we're looking at these sites throughout genomes and predicting whether or not they are in fact a splice site or just a decoy. So, and we formulate this as a binary classification problem, and that is the prediction.
So you want to find those very significant features that will be consistent given the species?
Yes.
Okay.
Yes.
Can you talk a little bit more about the algorithms that were used as part of the classifications?
The reinforcement learning algorithms?
No, the prediction about the splice sites.
The prediction. Oh, so we train 18 of popular, prediction algorithms like naive Bayes, SVMs, random forest, all these popular algorithms. We do that on backed versions of the data, and we end up with 180 models, right? So think about how you would put those together, right, to find a parsimonious ensemble that's also very predictive. So yeah, just any popular algorithm we train and get a model.
Yeah, so they are trained on different batches of the same training set? Is that how it works, or if all of them are trained on the same set?
So we have a training and a validation and a test data set. They're all trained on the training set, obviously, backed versions of the training set. We do some balancing also, again, because this is a very imbalanced data set. But yes, and in this case, for example, whatever those two models that are available at experiment 20 are also included in whatever available at experiment 40, right? So we do this incrementally. We keep adding and see how, for each experiment, how does the agent behave, what is able to capture, what are the final ensembles looking like.
I'm assuming they all have the optimal hyperparameters already available.
We use default parameters. We didn't want to get into that, right? We just use whatever is the default for that particular algorithm. Don't want to overfit. Yes.
Thanks for the great talk. Well, the term diversity is still a bit foreign to me. Like, I'm thinking about diversity as how similar are the algorithms are. Like, say, for example, well, I don't know much about the prediction, so I'm taking a general machine learning as an example. So, well, maybe the random forest ensemble algorithm is a bit similar to another ensemble-based algorithm, AWS, for example, and maybe SVM is similar to whatever. Is it
Sure.
the diversity one?
So diversity, we're looking at diversity in terms of predictions. How are the predictions made? Sometimes you don't have control of what algorithms to train or what data sets to use. Think about, for example, the dream challenges, right? Where every participant is allowed to train whatever algorithm they want on whatever data set they want. So you have, in this relatively uncontrolled scenario, you don't know what the models exactly look like. But you know the predictions that they make. So you want to be able to use models that are complementary of each other. Okay, so in terms of predictions, right, they want to be diverse. They want to be able to predict different things well, such that when you put them together, it boosts the overall ensemble prediction.